Welcome everybody to the NeuroTools Webinar of the week. Today we are joined by Dr. Armañanzas, who will be showing us the world of neuromorpho.org. Without further ado, I'd really like to hand it over to Ruben and allow him to get started, but we just wanted to thank you all very much for being with us today, and we're excited for an informative webinar.
All right, thank you, Devon. Thank you for having me today. I hope we're going to have some fun and learn some new things about our long-standing project, neuromorpho.org. I'd like to start by saying that I hold a faculty position at the Krasnow Institute for Advanced Science Study as part of the George Mason University. And that's the institute where the project is hosted and where we do all the work. I hold the hat of Director of Technology of the project of neuromorpho.org. So my background is in computer science, bioinformatics, and neuroinformatics. You can check my webpage there in the presentation, and let's start with the here we go with the outline for the presentation. So I'm going to talk a little bit about neuromorphology, and it's going to be like a brief intro about neuromorphology and why it's important, and we consider that it's a basic piece of basic neuroscience. What are digital reconstructions and how are they generated? I'm going to dive a little bit into the history of digital reconstructions, when everything started, and how off are we today. Then some not very seen graphics about the inner works of neuromorpho.org. I'm going to try to explain to everybody in broad terms how we work and what is the process from the moment we get data or a publication with reconstructions to the moment they are released in the web page. Then some lessons we learned from the 10 years of service to the community, including our take on data sharing in this field. I will highlight some of those lessons that I believe are important for other projects like us. Some future prospects that we are currently working on, and then I'll finish the webinar with a hands-on demo showing the main entry points for the repository that you can explore yourselves. And obviously this will be the Q&A part of the webinar, including any questions that I can answer to. All right, so in terms of the neuromorphology, I like to start by the what I call the three realms of the neuroscience, the neuron identity, and these are basic dimensions or realms of neuronal characterization. So we see here examples of morphology, physiology, biochemistry. These feature domains are highly interrelated with other fundamental aspects of neural identity, such as connectivity, development, and plasticity. Whether one believes in canonical cell types or in circuit or network doctrine, these realms, three of them, are intimately intertwined. And today, what we are going to explore is the morphology domain through our more than 10 years experience sharing digital reconstructions of neurons. So when we wonder about the diversity of neuromorphology, we can be facing some complicated task. You see here a figure from a paper from Pareg and Askley in 2013. And here you see neurons communicate, we know, through axons and dendrites, which are specialized tree-like processes. The importance of these branching structures is noted by their broad diversity across and within regions and species. So in here you can see a sample of cells from our repository, including just some examples like A, it's a Martinotti cell. We have pyramidal cells here in C and D. We have this K1, it's a spiny ganglion motor neuron. We have the typical Purkinje cell, and then this is this last one, the N, is a neocortex interneuron. So in red you can see the axon, and in black you can see the dendritic tree. So one thing is clear out of the diversity of morphologies is that the structure of these cells has a high relation with the function that they proceed. So when this tracing morphology started, and it started in 1888, in which Santiago Ramón y Cajal did his first reconstructions. So I want to stop a little bit and be a lot out of his script with Santiago Ramón y Cajal, because probably we all know about him. What probably you don't know is that he was a physician by training, and he served as a captain of the Spanish army in the first Cuban War for its independence in 1874. It was a short service. In 1875, he was sent back to Spain because of extreme malnutrition and possible malaria, although later in his life he had TB as well. So not an easy in terms of diseases life. However, he got to know the Golgi staining using silver in some years earlier of 1888 in a conference. And then he started exploring the options to stain the cells in the brains and inspect the results through the light microscope. So we already have the labeling, we have the visualization, and now the tracing, it was just whatever he looks down the microscope, use a pencil and draw it in a paper. No computer whatsoever. And that was the first reconstructions ever done with enough detail of the neurons themselves. So there are plenty of sites that you can check the drawings from Ramón y Cajal. This comes from a paper from the Felipe and Jones in 1992, and you can see how he started to not only draw single cells, but also put them into context and identify them and the possible connections between them as well. There are other drawings, very remarkable ones with different brain tissues, different layers of the neocortex. And eventually, he proposed the neuron doctrine, and he hypothesized that these cells were communicating using the axons and receiving the inputs to the dendrites. So he hypothesized and established the communication in the neuronal circuits with the pre and the post synaptic elements. Now, if you want to see some of these drawings, there is an exhibition going on right now in the US I believe in the US and Canada. I believe now it's in Vancouver, but it will go through New York, Boston, and North Carolina. I don't recall the city right now, but it's gonna go through all 2018. So check it out. If you have the opportunity to go in person and see these drawings, they are really remarkable, and you're gonna see that he was not only a scientist, but an artist, because these are handmade pencil on paper drawings. So what do we now? Well, what we do now is mostly the same tasks, but with a lot of help from the technology. In terms of the labeling, we can still do bulk loaded dyeing of tissues and see what are the neurons that are dyed by the dye. But we can also use intracellular staining or immunolabeling, or even we have genetic labeling in which we can pre-label the mice in this case, and genetically engineering so they express certain proteins, and then we can trace those proteins down the microscope. In terms of the visualization, we still have obviously the bright field microscopy, the light microscopy, classical microscopes, higher resolution with a confocal 2-photon microscopy, and even we can also go into the electron microscopy and reconstruct those stacks, as some groups are already doing. For tracing the cells, still there are places in which they are doing camera lucida reconstructions, basically just using like the shadow of the reconstruction and tracing that shadow with a pen-like device. There were some digitizing tablets helping this process as well. These are no longer in much use. Most of the digital reconstructions right now are produced using a semi-automated approach. That's the classical combination is a confocal microscopy attached to a computer with a software that is able to predict part of the trees, and then the researcher just has to either validate or correct the software and extend some of the branches that are a little bit dim, so they are not well recognized by the software. And hopefully at some point in the future we will get fully automated softwares able to accurately reconstruct the full structure of the cells from scratch. There are some prominent projects with this aim, and this is also part one big goal from brain technology. So the output of all this process, it's the reconstruction itself, which can be just the drawing, plain drawing of the branches and the soma, or we can have something as complex as this reconstruction, which will include elements as buttons, as spines, multiple channels, in tracing multiple compounds within the cell and the branches. We can have a full reconstruction of the soma with the 3D shape of the soma and so forth. So it can be from very simple reconstruction to a very complex with several elements included inside it. So what are the applications of digital reconstructions? Well, we can think on several of them. The first one, I already talked about it a little bit, is the neural identity. Can we classify? Can we define the different types of neurons just based on the morphology. The detailed characterization of the cellular anatomy is essential for elucidating also neuronal computation, for instance. And digital reconstructions not only increase the reliability of anatomical quantifications, but they also enable biologically realistic simulations for investigating the neuronal structure activity relationship, or the individual morphology, growth and theories behind it, or massive analysis of several hundreds of reconstructions trying to get the inherit rules from the reconstruction. So all of these are also interrelated between them, and there is a wide variety of cells that we can use for all these different applications. And there are several repositories and web pages that you can also browse and see what people are doing with the reconstructions. Now, moving ahead into our project, into the specific of neuromorpho.org, because the talk was called The World of NeuroMorpho. I would say that NeuroMorpho, in a nutshell, is a freely accessible and centrally curated repository of skeletons of axons and dendritic morphologies, and they can be traced from light and electron microscopy. We will see that we also have glia cells right now, not only neurons. So that's what we like to say, that it's skeletons of axonal and dendritic morphologies, not only neuronal morphologies. So the project itself, in terms of the technology components behind it, includes 3 main stages from the time we start our search for reconstructions to the time that they are released on the web page. Here you see broad workflow overview of how this process is done, starting with the literature search, in which every month we look into the neuroscience literature looking for papers that may include reconstructions. So that's what we call the reference evaluation, and then we identify which are the positives and what could be the preliminary metadata, which are the annotations of those reconstructions. From them, we release every month our results in terms of the positive and negative, and the exchange with the authors of those papers relating whether they are willing to share those data or not, or what is the status of the sharing for that data. Once we get the data set, then there is a pre-processing of that data set that I will cover in the following slide, including pre-processing and processing, also the metadata, final extraction, and then there are some other usage annotation and usage reports that we use for our internal consume. The second layer is the release of the data itself. So this is more IT related. What we do here is to grab the data and the metadata, change our database with a set of ingestion checks, and update the schema, do quality checks, release a preliminary version of the data into what is called the review side, and then that side is used to communicate with the authors and get their approval or their feedback, and open and have exchange a communication with them related their results. Lastly, the last stage is do the public release what's called the main site release. This is obviously taking all the content from the review once it's been approved and curated, and then we list all the new content and functionality. We provide fresh stats. We update our FAQs, whatever we need to do. Then we announce to the community some days later that the new release of NeuroMorpho is out. Okay, so this is the general workflow. With relation with the data files themselves, this will be the workflow. I'm not gonna go through it, but because we always release the original files from the data contributors and all the changes that we have done for the standardization of those files and our standardized reconstructed file, we have to go through this loop of normalization, check for things that are not okay, conversions. Is it ready to go? Is not ready to go? Do we have more issues with those files? Then we quantify using a measure the morphometrics of each of the cells that we're gonna include. We generate the images. We create all the files that we eventually release through the repository. The other big workflow would be the web release itself. Again, this is also purely IT work, and this is just a detailed view of what happens behind the scenes whenever we do a release of NeuroMorpho. And you can imagine that this is time consuming. The whole process usually takes up to 6 months, although during 2017, we are doing our best to have 3 releases within the year, and 7.3 is going on, and I believe that it will be ready before the end of the year, so it will be released sometime probably after the SFN or early in December. So here what we take is the metadata and the measurements coming from the cells. We ingest that into the database. We have our dataset server, and there is some communication from databases here. The literature release also. We generate the new content from the web page. We take the data and the image files. We send them to the release server. Then there is many pieces together that has to be put for the application deployment, and eventually there is a full communication between the release server, the public API that I will cover later in the hands-on, and also feedback from the authors and the users, and our tracking server, and new functionalities, internal wiki, and so forth. As you can imagine, this is a pretty large project, and we have a number of functions and servers going on behind the scenes. Okay, now moving into the figures or the numbers of data sharing in our experience, I'd like to show everybody how this business changed in between 2013 and 2014, mostly. You can see here all the evolution of all the versions, releases, pardon, of neuromorpho.org since the start in 2006 up to 7.3. This is still not officially released, but as I said, it's on its way, and it's going to be in 2017. So you can see the trend of number of reconstructions throughout these years. This accounts almost for 7 years, from more than 7 years, to 2006 to 2014. You see that this was, well, there was increase, but it was not big. Suddenly, the technology evolved, and then the slope of this trend changed drastically. And we kind of expect even bigger increments in the years to come due to all the automation that is happening in various different projects to acquire reconstructions in an automatic manner. So here we were in version 5.7 in roughly 11,000 reconstructions in total. And just after 4 years, we are already in 80,000 reconstructions. Okay, so what about the categories of this data? It's like, how complex are these reconstructions? Because we cover all the species and regions we can find. You can expect something similar in terms of the brain regions, cell types, species, and archives. And in fact, you can see more or less the same, although a species hasn't increased that much because, well, we know that we tend to work always in the same species, and it's hard to find a research group that it's going for a species that hasn't been worked on previously. Brain regions start growing again in 2014 and increase a little bit, so we're good. The number of archives is the number of labs that are sharing their reconstructions with us, and obviously we see also a peak in that, and we mostly saw a peak in cell types. Here's a disclaimer that I have to say, and it's like all these cell types are provided by the contributors of the data, so we are not labeling the cells, we are not classifying the cells. We have our basic types, which everybody agrees to, and then the more specific types for those cells are provided by the contributors, by the authors of those reconstructions. So we try to keep a balance with them, but we just put up in the repository whatever the author basically tells us to include. Now, these numbers are remarkable, but I also want to share that they could be twice as remarkable if people will agree more to sharing the data rather than what we are getting. So this plot shows us from roughly 2012 to 2017, almost up to date, what is the trend in our evaluation of papers, including reconstructions. So the green light that you see here is the number of reconstructions that are available through the repository. So the maximum here is 80,000, that's gonna be available soon. The previous one is 70,000 something, it's in here. But you can see this other red line going up of the green one, and these are reconstructions that we know that were done in papers in the literature, but for very different reasons we couldn't get our hands into them, so they are not available. Sometimes it's just because the paper was produced several years ago, and the researcher doesn't know where the reconstructions are, or the postdoc left the group and he took the files with him or her. I don't know, multiple reasons. But if you account for how many reconstructions are there, were produced, but are not available, and the ones that are available, we could be talking about twice the number of reconstructions if all these red ones will be available. Lastly, the blue line shows our exchange, our determining availability with the contributors, with the authors, that say like, well, how many of them were in negotiations towards the release or not, sharing or not. So what will be the impact in the field for a repository like us? Some more figures here to show you, to see that sharing the reconstructions, we believe that it's a win-win for both parts, for us releasing the reconstruction, for the user being able to download that information and including into secondary papers, into secondary discoveries, but also for the producers of those reconstructions, because we are a central repository. They don't need to worry about where those files are, what happened if somebody asks me for those files that I did 5 years ago. We will take care of that. We create the reconstructions. Sometimes we even detect errors that the original authors were not aware of. And there were some corrections because of that. And those are extra peer review corrections in your data, not only in your manuscript. So we have statistics about the number of downloads in 2007, and there were roughly 50,000 files were downloaded from the repository. This year, so far, we've got 2.5 million downloads from the reconstruction. From the repository, accounting for the whole span of time, these 10 years, it's 7.5 million downloads. Now, if we try to quantify how much work that could have been for a researcher, and we have a rough average of 23 hours for each, for producing a single reconstruction. Well, 100 people working on those 7.5 million downloads, it would take 1,000 years. And roughly, we estimate this as in $600 billion in labor that can be said that it was saved because they were already provided by the repository. Now, in terms also of the statistics about the branches and the cable, well, we can say that all these downloads accounts for 35 kilometers of cable, neuronal cable, and 700 million of branches in the different structures. Some more interesting facts: most frequently downloaded stats. Most neurons are downloaded 11 downloads per cell per month. Purkinje neurons so far are very famous. We have 10 downloads per cell per month. And obviously neocortex is the hot topic in the field, so it's the hot topic also for us with 11 downloads per cell per month. Now, I'd like to move a little bit into our lessons learned from all these years of experience. The first point I'd like to stress is that we serve the end users. So we serve the researchers downloading the files and their work. So we always try to complement rather than duplicate existing resources. Another important point for us is to avoid proprietary formats as much as possible, and that's why we standardize and release all the reconstructions in a text-based format, the SWC, that anybody can open and all the softwares can read and interpret. Second big point is that let's make it easy for the data contributors. They already done the work. They had their paper published. So it's very easy to put the work on their end, whereas we should be the ones putting the work in our end when they want to share something. We always publicly acknowledge the data creator and the paper from which every single file is coming. we look for a balance in the metadata information we release, trying to be concise, consistent, and specific at the same time. Sometimes this is not an easy task, sometimes it's more easy. And the third point I like to stress is that we always keep the eyes on the data, as there is a lot of work for updates and maintenance of whatever it was released previously and whatever we're releasing in the future. We must have a diverse expertise within the team, because we are covering neuroscience, but we also have hot topics, hot technologies in IT, so we have to be on top of everything, and there is an automation, and if we want to scale up to the pace of production, you need to have a very diversified team. So these short points and many more were published in Nature Methods this year, and the paper is called Win-Win Data Sharing in Neuroscience. If anyone wants to check more on that. So wrapping up in here, future trends, we expect to publish multi-channel reconstructions and time-lapse information. These are some examples of this. This would be not only including the branches, but including the evolution and the presence or absence of certain molecules in the different branches. As I said, we already have some reconstructions from glia cells. This is one example. And we are acquiring more and more, and we are releasing more and more. Now, for the far away future, we expect, or we estimate, that the year increments will be around 20,000 cells per year, but you never know, because there might be a new breakthrough in a technology that put this number into higher figures. Another front we are working in is the automatic detection of positive publications. So far, we have a semi-automatic detection, but it has to be corrected by a human. So we expect to have this done automatically, in the sense that we scan one paper and we say with some artificial intelligence that that paper included reconstruction and it's a positive, or no, it's not a positive. And lastly, we know that the cells in the repository are floating in space alone, so it's hard to register them in a shared space to do more connectivity analysis. So we are working on that front as well, to do a co-registration of the cells that we publish, or a sort of a subset of them within known atlases. All right, so acknowledgements. I like to thank, as you can imagine, a large team. This is the current team members. We have Giorgio Ascoli, which is our principal investigator, and then there are 17 people actively working within the project right now. We have PhD students, we have visiting scholars, master's students, undergrad students, and some volunteers as well. The support, it's all academia, so coming from taxpayers, NIH, NSF, MURI, Keck. We had some collaborators for very, very punctual tasks, the Mitre Corporation, and we have people working on CDAP, NeuroML, and the Allen Institute as well with the Brain Explorer. But I also like to thank all of our former team members during these 10 years. I tried to keep a track of a record of everybody working on the project, and so far there are 56 former members at the different levels for this project, and we are all very grateful to all of them for their work and for making us reach the positions that we hold these days. So this is the end of my presentation, and I'm gonna hand over to you, Devon, for possible questions on the presentation.
Yeah. Great, thank you very much. It looks like so far in the chat we don't have any questions about the content so far. However, if anyone would like to unmute themselves at this time or type a question into the chat before we move into the demonstration, feel free to do that.
No, I would like to thank you very much for this very informative talk. I mean, I think we've had a couple of demos of NeuroMorpho before, and but what you did is kind of cover the field in a very deep way and an understanding way. And I just wanted to know what your feeling was in terms of, so why are we getting so many more neurons, neuron reconstructions? Why has this increased? And I was actually looking also at our statistics in DISCO. Can you kind of think about and chat about what is the reason why we're getting so many more? Is there are there some journals that are now pushing this? Is it just a cultural shift? Why are we getting now so many more neuronal reconstructions, other than what you did say, which was now there are more tools to do it? But is it all tools, or is it culture? What do you think? Or is it your wall of shame? That's the other question.
Very, very good question, Anita. Well, I think it's a combination of both factors plus some other factors. I think there is a social factor involved in this, and it's just a natural evolution of the field. Back here, I'm already in the page neuromorpho.org in the About that you can access from the home About. So you can see the dynamic version of the plot I show with the numbers for each of the version if you browse over the different numbers. So I believe that there was an evolution of the labs in terms of postdocs learning how to do reconstructions, and then moving into PI positions from the very famous labs, and then starting their own lab and also doing reconstructions for other kinds of neuroscience questions, of course. But that took some time to ripe, and then I think that the combination of that, in addition to a more open-minded regarding sharing the data and their work, really boosted the evolution in here, the numbers. And it was also backed up by the fact that the confocal prices of the microscope, the prices of the confocal microscopes were a little bit lower, and there were more money. The prospects of the Brain Initiative and all the big projects from Europe also boosted the number of reconstructions. And what I'm seeing these days is that there is also a new trend for some traditional labs to say that I don't want to do more reconstructions anymore. I'm expecting to have an industry-based facility that is going to produce the cells for me at a very affordable price.
Excellent. We also have another question in the chat from Dr. Pollock. Can you explain how this will integrate with Brain's cell census? That's capital BRAIN lowercase s cell census.
Yeah, well, what we do in terms of integration with other projects is, first of all, open the conversation and the discussion on how to do it. And obviously for each of the cells, we have the cell types that we can think about the cell census and the, okay, what are the classification of the neurons? What are the types? We are willing, and we are in open conversations with other repositories to include links for some given cells that we all agree upon to interlink our repository with that, with those repositories, and exchanging that information basically, and providing the users with the ability to browse our categories and the other categories, other people's categories as well. And so that's basically how we do things, is work in a team manner with whoever is interested in just exchange knowledge and create what we call a knowledge network.
Excellent. Thank you, Ruben. I think we are ready to move into the walkthrough. It looks like our questions have been answered.
Okay. So this is the main page of the entry page of NeuroMorpho. It's neuromorpho.org, obviously. Here in the tabs, you can see the main entry points. In the home, there are some basic information that some of it I like to cover. Then we have the browse and the different browsing options to dive into the content we have. Then there are also several search opportunities for the savvy user to search the repository and our resources. There is the third tab, it's the literature coverage, in which we every month publicly put up all the papers that we have searched and what is the status in terms of sharing of those reconstructions of those data with us. Then there is a terms of use, and there is a help with some useful links that if I have time, I'll also like to cover some of them. So one important bit from the home to me is the what's new. I was referring to this before. The what's new page, it's everything that happened in neuromorpho.org since its start. So let me go all the way down. You go all the way down of this long page, you will see that this was the very early start of NeuroMorpho, in which you can see what was included in the beta release in the 20th of September 2016, 2006, including only 1,000 neurons. So from the get-go, we've been covering every single edition and keeping a record of all the changes and all the new archives and cells that were included throughout these 10 years. And every new release, we got a listing similar to this. The first thing is the new archives and how many reconstructions with direct links to each of those reconstructions in a summary way. And we also include how many new species, what are these new species, what are the new strains, whether we have new brain regions and which are these brain regions, cell types, experimental conditions, original formats, thickness values, stain softwares and stain strains, and other changes that we might have done to the reconstructions or to the functionality of the repository, including updates to the FAQ as well. So this is one important anchor to browse and to check if your favorite lab has some cells that you want to check. You can check in here. Or you can go in browse and go by browse by archive. This will load a pie with the, let's say, big guys, with the archives that have pretty large number of reconstructions with us. But you can also see the full list of all the labs that gratefully share data with us. Because this is long.
Excuse me, Dr. Armañanzas. Sorry to interrupt you, but I know you said we could jump in at this time if there were questions in the chat during the walkthrough. Cool. So we do have a question in the chat from Lim Jing Xuan. Is there already a repository for channel and synaptic densities and other molecular information for the cells reconstructed in NeuroMorpho?
I cannot answer 100% sure to that question, but I don't believe that every single lab will do all that level of detail for each reconstruction, because many cells might be in culture, or they might be the paper producing those cells maybe looking for a specific change in phenotype. So, for instance, that it doesn't require them to record that detailed information. They just need to record the branching structure. So they don't go through it, and hence there is no such information. So we have what we have. We have what the contributors produce in the papers, and I would say that the best way to know if we have the original files with that information is to browse through the literature and see whether those papers include those elements or not. In terms of our search abilities, we don't have integrated that yet. All right. So here you can see this, I was saying, this little search box, and you can just type Markram, for instance, you'll get all these reconstructions are the ones included in the Blue Brain Project paper. When you browse over them, you can see a preview of the reconstruction to your right. If you click in any of them, you'll go to a page similar to this one. It's the detailed information for a reconstruction. And here you can find the morphology file, which is the SWC standardized by us, the original morphology file produced by the paper, and then 2 more log files accounting for the changes that we've done from the original file to the standardized file. Now there are a couple of more links here, the 3D neuron viewer and the animation. The animation opens a pop-up with GIF, including an animation of a possible growth from the soma to the more distal branches of the cell, and then it goes again from the soma to the branches. Now, if you click on the 3D neuron viewer, you may need to give Java some security clearances to run this software. It's just basically a CV app. Oh, this one's not here. Okay, well, that can happen. Let's try another one. Yes, here it is. Okay, so here with CV, this modified version of the CV app, you can browse the cell, go in to zoom, inspect the structure of the dendrites and the axon in this case, center it back again, going, zooming in, zooming out. And here there is the option to export this file in other formats, NeuroGenesis, NeuroML, different versions. Okay. Now, down in the page, you'll get all the metadata information from that cell, and also the referent articles from which that cell was collected, and the measurements, the morphometrics of this cell in particular. The other big entry point for NeuroMorpho, besides the browse, which can be done by animal species, brain region, and cell types, and archive, is the search. And here in the search, you will find different searches. One is done by the metadata or multiple neurons. And here you can include whatever animal species, for example, gender, weights, ages, experiments, protocol, all the metadata is searchable. Also the brain region, the cell type, the source of the reconstruction, and the completeness, which is something that we included relatively, not a long time ago. A structural domain, you can look for dendrites and axons and soma, or if you're only interested in dendrites and you don't care about the soma or about having an axon or not, so there are multiple combinations of these. And you can also look for processes that are not labeled as dendrites or axon. Physical integrity, you can look for dendrites and axon, neurites and processes. You say neurons, search by dendrites and axon, then let's say what is labeled as complete. Click on the hits. It should give us a number of what is the total of cells that we have labeled as complete for dendrites and axon. In this case, is around 1,100 of them, and then you can display them by animal species, brain region, cell type, archive, or just show a summary of all of them. And this may take a little bit of time because of here we are. So it's retrieving the neurons from the database. So the pagination is based on 500 cells per page, and that's why we're getting 3 pages, and eventually it will show the summary of the first 500 cells. Before I run out of time, I like to also stress this other recent improvement in the repository that we included, and it's the establishment of our own application programming interface. So we have already a public API, which is a REST platform using the HTTP as a communication layer. If you go to neuromorpho.org/api.gsp, then you'll get the information on how to run the API and do the queries to the API. The entry point for the API is very simple. It's just neuromorpho.org/API, and this is the health page showing that it's working well, and it has 3 main anchors to retrieve data: neuron, morphometry, and literature. I have to say that this is currently in development, so it's stable enough to use, but we are including more and more refinements from one version to the following one. But this is important because it allows people to do massive queries to the repository, getting all the metadata information, or the literature, or the morphometry values for the neurons. And here we have the 500 reconstructions, the summary of them that are labeled as dendrites and axon complete. So you can see some very nice cells with huge axons. This one from the rat, it's the hippocampus CA3C, and this is a pyramidal cell. And I think that I'm running out of time. So I don't know, Devon, if there is any other questions that...
It looks like all of our questions have been answered at this point, Dr. Armañanzas.
Okay. Well, I think that was roughly it. I could spend easily another hour explaining all the particulars of browsing features of the repository, but I will encourage everybody to just play around and look for it, because there is a lot of hidden content in the page. People can also look into the help and tools and links, and then you can see here a list of tools related to neuronal reconstructions and some other web pages and other resources. We have here an updated table of resources and tools for 3D reconstructions of neuromorphology. And here there is even more: the resource, the platform, the support, the reference, and the web address. So anybody interested in the field, it's very welcome to browse the repository. Write to me with any doubts or comments, and I'll be very happy to answer them.
Well, great. We'd just like to thank you very much for this informative webinar. You're getting thank yous from the group chat as well. Thank you from Dr. Bandrowski as well. We would like to encourage everyone to share this webinar with their colleagues. We will be posting it on our social media channels in about an hour or two once I run it through our Adobe software. On a personal note, I'd like to thank Dr. Armañanzas for his patience with my pronunciation and just for an excellent webinar. We hope to see you all next week as well for our next live webinar. On that note, I think I'm going to end the meeting, unless there's anything else you'd like to add.
No, I would like to thank you guys, Devon and Anita, for putting up all this series of webinars. I've been watching the first one. I was live, but the others I've been watching them through the YouTube channel, and I believe that this is a really good class that you're doing.
Thank you. And we have one more question from Dr. Pollock in the chat. What is the URL for the posting? I think that's in regards to the YouTube link for the recorded webinar. That will be through our YouTube channel, and we will post that link once it is created on our upcoming webinars website, which is newinfo.org/about/upcoming webinars. I'm going to put that into the group chat right now. And if you scroll to the bottom of that link, you will see all of our YouTube links for past webinars. After today, this webinar will go to the bottom of that web page, and it will have the YouTube link on it. Just a little logistics information.
Thank you so much.
Thank you to everyone who joined. Thank you again to Dr. Armañanzas, and we hope to see you next week.